Awesome, thank you. Perfect. And can people see my screen and my cursor? Fantastic. So yeah, as she said, my name is Kevin Guttenplan, and I'm a PhD student first in the lab at Ben Barris, and now finishing up my work in the lab of Aaron Gittler. And I'm going to talk to you guys about my PhD work, which has been trying to understand the role of reactive astrocytes in disease and injury. So astrogliosis, or an astrocyte becoming a reactive astrocyte, is a pretty generic term for this idea that astrocytes seem to respond to pretty much any insult to the central nervous system. So if you stain for an astrocyte using gliofibrillary acidic protein, GFAP, which stains most astrocytes, and you're looking like the kind of quiescent, happy brain, they have this nice ramified morphology, thin wispy processes that gave them their name of star-like cells. But if you start to have insults to the central nervous system, in kind of a moderate insult, they'll start to hypertrophy, so they'll swell up their cell bodies and their processes. And in really extreme insults, like a stab wound or a stroke, they'll start to proliferate, they'll grow on top of each other, lose their normal morphological features, and form something called the glial scar, which is thought to kind of cordon off the area of injury. So we've known that this response seems to be occurring for a really long time because we've seen these morphological changes, but because we've been mainly tracking these morphological changes in the expression of this one gene, we haven't really known the functional changes that astrocytes might be undergoing. The first kind of hints we had that there might be interesting functional changes that are occurring is when people started looking at mouse models of different injuries, so stroke, neuroinflammation, often modeled by LPS injection. LPS lipopolysaccharide is just a bacterial wall endotoxin that when injected kind of simulates a large-scale bacterial infection. Spinal cord injury and then in neurodegenerative disease models such as mouse models of ALS. By using kind of crude methods for manipulating astrocyte reactivity, they started to notice that getting rid of reactive astrocytes had beneficial effects in some contexts and detrimental effects in others. So for instance, in like ALS, it suggested that astrocytes secrete something that kills motor neurons. But in spinal cord injury, if you get rid of reactive astrocytes, axon regeneration is impaired. So that would suggest that maybe the astrocytes are normally doing something beneficial. So the fact that astrocytes seem to be doing, having grossly different effects in different diseases suggested that not only might they have interesting functional contributions to these diseases, but also that they might be kind of specific to different diseases and injuries. So in order to kind of attack this question of what these reactive astrocytes might be doing, Shane Lidlow, who's now a PI, but at the time was a postdoc in the Barris lab, and I tried to understand the mechanisms and roles of astrocytes in one specific injury, which was LPS-mediated neuroinflammation. So what we found in studying this particular kind of response of reactive astrocytes in neuroinflammatory contexts is that LPS seemed to come in and somehow activate microglia. Those microglia then secreted three factors: IL-1 alpha, TNF alpha, and C1q. And those three factors together were necessary and sufficient to induce this neuroinflammatory reactive astrocyte response. And what's really cool is we could then use the nice quiescent serum-free astrocyte cultures that the Barris lab developed in order to study the response of these astrocytes by just adding recombinant versions of these proteins and really specifically study the functional changes that they underwent and how they changed the interactions with other cell types. We also generated an IL-1 alpha, TNF alpha, C1q triple knockout mouse so that in vivo we could stop the signaling pathway and stop the formation of neuroinflammatory reactive astrocytes. Using these two tools, the kind of functional changes that we generally found occur in neuroinflammatory reactive astrocytes are: one, quiescent astrocytes normally promote synaptogenesis. There's been a lot of work in the Barris lab over the years characterizing how astrocytes induce the formation and maturation of synapses. And these reactive astrocytes no longer promote synaptogenesis, and they may actually actively disassemble already formed synapses. Astrocytes normally phagocytose synapses in myelin, so they participate in the kind of normal developmental synapse elimination that is kind of most famous in microglia. And these astrocytes decrease their phagocytosis, both in vivo and in vitro. And then finally, and probably most intriguingly, astrocytes have long been thought to be kind of essentially neurotrophic. So in vitro, astrocytes are often required to have healthy in vitro neuron cultures. And in vivo, it's also believed astrocytes are required for normal survival of neurons. But these reactive astrocytes actually were actively neurotoxic. And we found evidence that they secreted some factor or set of factors that actively killed neurons and oligodendrocytes, but not other cells of the central nervous system. So all these functional changes together led us to the question, which is the kind of the basis of the basis question for the two papers that I'm talking to you guys about today, which are: do these neuroinflammatory reactive astrocytes contribute to disease and injury? We now know that they undergo these interesting and specific functional changes from our work in vivo and in vitro, but we wanted to know, do they actually have any impact in central nervous system injuries and diseases? So in order to answer this question, we wanted to first start with a very simple injury. And so the simple injury that we came up with is an optic nerve crush. So what you do is you come in and you crush the optic nerve just behind the eye, and that crushes all the axons of the retinal ganglion cells that project from the eye into the brain. So if you dye fill these retinal ganglion cells after crush, you can see that you see these coursing axons going into the optic nerve, and they all stop right at this crush site. So what normally happens after exotomy of these retinal ganglion cells is if you just stain the retina for retinal ganglion cells here in green with RBPMS, which just stains all retinal ganglion cells, over the course of about a week to a month after crush, you lose the vast majority of retinal ganglion cells, shown here by the lack of these green cells. So you can quantify this in a normal wild-type mouse, and you can see that you lose the vast majority of retinal ganglion cells over the course of about a month. Interestingly, if we did the same insult in the I1 alpha, TNF alpha, C1q triple knockout mouse that we generated that stops the formation of neuroinflammatory reactive astrocytes, we no longer got the death of those retinal ganglion cells. Further, we were able to recapitulate this in a kind of a more therapeutic way in rats using, instead of knocking out the 3 genes, we used neutralizing antibodies to inhibit the signaling of I1 alpha, TNF alpha, and C1q by injecting those neutralizing antibodies directly into the eye. And again, by stopping that signaling pathway to astrocytes, we're able to completely stop the death of retinal ganglion cells. And so this suggested to us, similar to what we saw in vitro with astrocyte toxicity, this microglial astrocyte signaling pathway seemed to be pretty important for the normal death of retinal ganglion cells that occurs after optic nerve crush. So this is interesting, but we wanted to move into a slightly more complicated and slightly more neurodegenerative disease model. So the next model we looked at was a microbead injection model of glaucoma. So in this model, you inject microbeads into the eye, plugs the aqueous outflow of the eye, and that causes an increase in interocular pressure, which can be shown here in this red, beginning over the course of a week, but extending to about a month. And what normally happens in this prolonged increase in interocular pressure model is you get the death of retinal ganglion cells, shown here again by staining for RBPMS in the retina, and you can see a slight decrease in the number of retinal ganglion cells over the course of a month. And again, like what we saw in the optic nerve crush model, we're able to completely stop that death in the triple knockout mice that lack the ability to induce reactive astrocytes. And you'll notice that compared to optic nerve crush, it's a slightly less dramatic model, but if you look at SMI32 positive alpha retinal ganglion cells, which are particularly sensitive to this insult, you can see they normally lose about 50% of that particular subset of retinal ganglion cells, and again, we're able to completely stop the death of that subset as well. Now, this is really interesting to us, but the other thing we wanted to investigate using this model is there's a lot of ways to stop the death of a cell that don't necessarily leave the cell functionally relevant to the circuit that it's been removed from. So, you might be able to stop the final execution or caspase of a cell death cascade, but if all the upstream signaling cascades have occurred, sometimes the cell will be so irreparably harmed that it's really not that functional anymore. You basically just saved the husk of what was once a cell. It just hasn't technically died. So in order to understand whether that's the case, we wanted to look at what are the morphological and functional changes that have occurred in these cells that we've now stopped from dying. And again, what I'm going to show you is the kind of standard of comparison is we're not really comparing it necessarily to are these neurons normal, but compared to a normal neuron that's dead, are they better than being dead? So the first thing we want to look at is their morphology. So we dye fill retinal ganglion cells. You can see here in a control uninjected triple knockout retina, you compared to the triple knockout retinas with an increase in interocular pressure. You can see that in general, their morphology is somewhat reasonable. Their dendritic arbors are a little bit more compressed compared to a normal unaffected retina, but generally speaking, it still looks like a retinal ganglion cell. We can skeletonize these in order to show this. So again, this is looking top down. You can see that same thing from over there with a slight decrease in radial dendritic arborization. Interestingly, if you look at the side view, there's actually a bit of an increase in the width of arborization. So normally these kind of super happy retinal ganglion cells, their dendritic arbors lived in a very specific lamina of the retina, and these slightly injured ones slightly increase their arborization of the dendrites within that axis. But in general, their overall morphology is somewhat preserved. And because the overall morphology was somewhat preserved, we wanted to know, is their function still preserved? So we decided to turn to electrophysiology. So first we patched on to either the control retinas or these increase in intraocular pressure retinas. And using a current injection, just tried to drive firing of these neurons. And what you can see comparing a control to these being injected is that certainly the electrophysiologically properties are changed. There's a decrease in overall spike rate, but they also do have kind of the general same spiking behavior as a normal retinal ganglion cell. And more importantly, we wanted to know what is their function in a kind of more physiologically relevant context. So we did an increase, we exposed the retinas to an increasing size of light. So you start activating the retina with more and more and more light. And that means that the retinal ganglion cells are going to be summing more and more activation from the activated photoreceptors. And so what you would normally expect to see, which is shown here in gray in a control retina, is this sigmoidal curve where with increasing spot size diameter of light, you get an increase in firing rate until you reach kind of a max firing rate. What you see in these injured retinal ganglion cells that we've saved from death is that indeed, like what you see in this example to the left, there is a decrease in overall firing rate at kind of normal physiologically relevant light stimulation sizes, but eventually they actually are able to catch up. You still get this nice sigmoidal curve, and at kind of high activations of light, they actually are somewhat similar to uninjured retinas. So all this suggested to us is that while these retinal ganglion cells are certainly changed, in general, their morphology and electrophysiological functioning is somewhat reasonable, and thus maybe with some prodding and some other interventions, we might be able to actually make these functionally relevant to the retina and kind of helpful for the regeneration of the retina after this kind of injury. The other thing that we noticed doing these experiments was something that we didn't expect, which is apparently when you do this model, sometimes you get this increase in intraocular pressure that we talked about before, but it's only transient. So something happens, like the beads that are normally plugged in the aqueous outflow get dislodged, aqueous outflow returns, and the intraocular pressure decreases back to wild type after only a brief increase. And what's really interesting is if you take a bunch of markers that we use to mark these neuroinflammatory reactive astrocytes, and you average how much upregulation of those markers you get in either the normal sustained increase in IOP mice or these transient increase in IOP mice, what you see comparing in the retina here in the sustained versus transient is actually a very similar response of astrocytes, a very similar induction of reactive astrogliosis. And the reason that that's weird is that even in wild-type mice, when you have a transient increase in intraocular pressure, you don't get the death of retinal ganglion cells. And so what this would suggest is we have an instance here where you still get the robust induction of astrogliosis as shown here, but even in a wild-type mouse, you're not getting the death of retinal ganglion cells. That suggested to us that the toxicity of astrocytes is slightly more regulated than just the presence of reactive astrocytes or not. So in order to understand that mechanism more fully, we went back to the optic nerve crush model for technical reasons. And what I showed you before is that in a wild-type mouse in an optic nerve crush, you lose the vast majority of retinal ganglion cells over about the course of a month. And again, what I showed you before is that in the triple knockout mice, which lack the ability to form reactive astrocytes, you no longer get the death of those retinal ganglion cells. So in order to understand the kind of control of astrocyte toxicity, the next experiment we did is we took in vitro astrocytes and we made them produce the toxic factor that we found in vitro, and we purified that toxic factor, and we injected it directly into the eye of either wild type or triple knockout mice. What you can see here is there's no death of retinal ganglion cells. So even if we inject purified astrocyte-secreted toxin into the retinal ganglion cells are totally fine. But if you take these triple knockout mice that underwent an optic nerve crush, so their axons have been injured, but they don't normally have neuroinflammatory reactive astrocytes, so retinal ganglion cells don't die. And on top of that, we inject our purified toxin. We're able to restore the death of retinal ganglion cells back to wild type levels. And so what's suggested to us is that there's kind of a two-key model of regulation of retinal ganglion cell sensitivity. First, the neuron has to be susceptible to astrocyte-mediated toxicity, presumably by some sort of injury that in this instance is caused by the exotomy. But then secondly, you have to have the astrocytes produce the toxic factor. And so if you're missing either of those two key mediators of death, the retinal ganglion cells are totally fine. So this was really interesting to us and gave us a better understanding of the mechanism by which astrocytes might participate in these diseases. But again, we still wanted to get towards a more neurodegenerative disease model. And in that previous paper, that first paper where we characterized these cells, we also found that there was this marker, complement component C3, that in astrocytes was a pretty good marker for these neuroinflammatory reactive astrocyte response. And we took an antibody against C3 and looked in human patient tissue from patients with a range of different neurodegenerative diseases, Huntington's disease, Alzheimer's disease, ALS, Parkinson's, also multiple sclerosis, which isn't shown here. And what we found in general is that there seemed to be an increase in this neuroinflammatory reactive astrocyte response in most neurodegenerative diseases that we looked at. So what this suggested to us is that we could probably look at a range of different mouse models of neurodegenerative disease. And what we ended up deciding to look at was ALS, or amyotrophic lateral sclerosis. So what is ALS? ALS is a progressive motor neuron disease that causes the death of the upper and lower motor neurons that translate motor intention in the cortex into actual muscle activity in the muscles. And so in line with the death of those motor neurons, you lose, it's a progressive paralysis that usually progresses from the distal parts of the body towards the central parts of the body. And it's just an absolutely devastating neurodegenerative disease. It's more common than I think most people think, and it's really rapidly progressing, with the average date to death occurring as early as 14 months after diagnosis. So it's really quite devastating and more common than I think people think. And so like most neurodegenerative diseases, most people that get it have sporadic etiology, so they have no known familial history of the disease, but there is a subset of patients with known familial inheritance patterns, and we often look at those patients and try and identify mutations that might be causative for the disease to understand it better. And in ALS, the kind of first familial mutation that was found to be causative in ALS was SOD1, or superoxide dismutase 1. And if you take the mutant human version of SOD1 and you overexpress it in mice, you get progressive motor neuron degeneration and paralysis, somewhat reminiscent of the disease. And it's one of the most long-studied models, not only of ALS, but kind of neurodegeneration in general. So first, in order to understand if neuroinflammatory reactive astrocytes contribute to this disease, we needed to know if they actually form in this disease. So again, we went back to that marker, complement component C3. And in this instance, we used in situ hybridization, and we saw a dramatic upregulation of this neuroinflammatory reactive astrocyte response in the spinal cord of these SOD1 mice compared to wild type. And if, kind of like what we did in the optic nerve crush models, if we breed in the IL-1 alpha, TNF alpha, and C1q triple knockout mouse, which lacks the ability to form these neuroinflammatory reactive astrocytes, we're able to really dampen down that response in this model. So we've gotten rid of these neuroinflammatory reactive astrocytes. What does that actually functionally do to the disease? And much to our surprise, it actually really dramatically extended the lifespan of these mice. This is a very aggressive neurodegenerative disease model. Most mice died about 130 to 150 days, shown here in red. And the triple knockout SOD1 mice, shown here in blue, live about 2 months longer, or about a 50% extension in overall lifespan, which is a really dramatic extension of lifespan in this model. Now, there's two big ways we could have accomplished this extension. In general, we could have had the mice get sick later, so delayed the onset of the disease, or we could have slowed down the disease, so slowed down the progression of the disease, so the mice get sick more slowly. In order to figure out which of these two occurred, we used rotarod analysis in order to more carefully track the motor performance degeneration of these mice over time. What you can see here in red for normal SOD1 mice is there's a really rapid degeneration of motor neuron performance before endpoint. In the triple knockout mice, what we see is that they actually start to show overt motor phenotypes at a really similar age, but those overt motor phenotypes progress much, much more slowly over time. And so what this suggested to us is that we haven't delayed the onset of the disease, we've slowed down the progression. Now, the main pathophysiological correlate of this motor neuron performance is neuromuscular junction denervation. So before motor neurons die, they denervate from the target muscle. So we wanted to know if we've kind of slowed down that pathophysiological process at all as well. So we, in order to assess neuromuscular junction denervation, we used confocal analysis, staining the postsynaptic side of neuromuscular junctions with alpha-bungarotoxin to stain acetylcholine receptors, and staining the presynaptic innervation of the motor neurons with neurofilament. And you can see in a fully innervated neuromuscular junction, there's a really nice apposition between this presynaptic and postsynaptic staining. In a denervated neuromuscular junction, which happens increasingly as ALS progresses, you start to lose that presynaptic innervation, even if the postsynaptic staining still resides. And in really extreme instances, eventually the neuromuscular junction just disappears altogether. So if we track this denervation over time, you can see, again, comparing red to the blue triple knockout mice, we're also able to delay this neuromuscular junction denervation, again, by about 2 months, which aligns well with the overall extension of lifespan. Now, this is really interesting, but given the fact that these reactive astrocytes actively kill neurons, which I mentioned earlier, the other really big question was, did we in any way affect the motor neuron death? So in order to track motor neuron death in this model, we did in situ hybridization against choline acetyltransferase to stain motor neurons in the spinal cord. And quantifying this and zooming in here on the lumbar section of the spinal cord where most of the activity occurs, you can see that at the end point of a normal SOD1 mouse, you get about a 50% loss of motor neurons. At the same age of these endpoint, the triple knockout SOD1 mice have actually not lost any motor neurons left yet. But about 2 months later, when these triple knockout SOD1 mice do eventually reach endpoint, you do get about a 50% loss in overall motor neuron number. So what this suggested to us, kind of aligning with the other pathophysiological results and the overall lifespan extension, is we slow down this disease pretty dramatically, but eventually, once even these mice that lack reactive astrocytes do get full-blown ALS, they seem fairly inseparable from the normal progression of the normal model of ALS. So altogether, what this makes for us as a model for how astrocytes participate in this disease and maybe diseases in general is that there's some sort of initial injury or insult that causes the degeneration of motor neurons. So in this instance, it's this expression of SOD1 G93A, but in other, in human patients, it could be that, it could be other mutations, environmental insults, we don't really know. Once this initial generation of motor neurons has occurred, this is sensed by microglia and induces the conversion of astrocytes to this neuroinflammatory reactive astrocyte state. And then by some combination of the things that we found earlier that reactive astrocytes do, or maybe other functions that we just don't even know yet, that somehow induces the accelerated degeneration of these motor neurons. So in our model, by stopping the conversion of these astrocytes and stopping the participation of these other functional changes, we're able to not stop, but slow down this overall motor neuron degeneration. Now, this is kind of interesting to us because it resides as an interesting potential therapeutic target because most patients, as I suggested before, are sporadic in ALS, and as well as most neurodegenerative diseases. So that means that they usually come to the clinic with already showing phenotypes, already showing changes in their physiology, and thus the onset of the disease has already occurred. So very few patients are familial, and so there's very few patients that you can really catch before they start to show symptoms. And so if we were able to target this astrocyte reactivity response, that actually might be broadly beneficial to a lot of these patients that just come to the clinic already with symptom onset. But in order for that to be the case, we need to know that this reactive astrocyte response occurs generally in ALS, so it occurs in sporadic cases as well as familial cases. So in order to answer that question, we went back and again used staining against that marker complement component C3, and we looked for that complement component C3 response within GFAP positive astrocytes in either non-neurological control patients, ALS patients with sporadic etiologies, or SOD1 or C9 or 72 associated familial cases of ALS. And what we found is that in both sporadic and familial cases, and most extremely in the distal parts of the central nervous system where pathology is worse, we do get the general response of this astrocyte reactivity response. So we see it high in both sporadic and familial cases, highest in the spinal cord, kind of high all the way throughout the central nervous system, but lower as you get further away from the main pathology. Adding all these together, the things that I've shown you today is that neuroinflammatory reactive astrocytes seem to contribute to a wide range of insults and diseases of the central nervous system. So in an axon crush model, we can see that you get the death of retinal ganglion cells, and we're able to stop that by stopping this response. In a glaucoma model, we're also able to stop the death of retinal ganglion cells and actually preserve at least some of the normal morphological and electrophysiological functioning of the neurons that would normally die. In a mouse model of ALS, we're able to kind of quite starkly extend the overall lifespan of the mouse. And we found that this response seems to be generally applicable both in sporadic and familial cases of ALS and in many neurodegenerative diseases in general. So all of this work was helped greatly by the fact that I was able to work with a lot of different labs over my time in graduate school. So first in the Barris lab with special help from Maya Weigel as well as Alex Munch, but then also the Lidlow lab once Shane went and started his own lab, as well as the Gittler lab who became my new lab home after Ben's unfortunate passing, and then also some further work not shown here where I got help from the Chopper lab. And I'm now happy to take any questions you guys might have.
Great job, Kevin. Really interesting stuff. There are a couple of questions here. I'll start with the first one. What is the morphology of an optic nerve astrocyte that has become an A1?
In general, we didn't do a really close analysis of the morphological changes. And in general, we've seen some really interesting differences between the kind of traditional astrocyte reactivity that often occurs in, say, like a spinal cord injury and some of these neuroinflammatory reactive astrocytes. So at least in vitro, they don't really undergo the same hypertrophy and morphological changes that a lot of people report. And in vivo, sometimes they don't even change some of the traditional transcriptomic signatures that you'd expect. So sometimes you can get a pretty obvious neuroinflammatory reactive astrocyte without the broad upregulation of GFAP, which is often used as a marker for it. And so what this suggests to us, it's just meant that we haven't focused as much on any single marker or any morphological changes, just because they can sometimes be unreliable indicators of reactivity, whereas using a broad range of genes tends to be a little bit more of a careful analysis of it. So short answer is, I don't know.
So I have another question. So other than projection of LGCs, do you see optic nerve regeneration in the triple knockout mice?
Yeah, we were really excited. That was in my my quals back when I was an earlier PhD student. I was very excited about the idea that after saving these retinal ganglion cells that they might regenerate. And the short answer is it doesn't seem like they do. We didn't do an exhaustive analysis, but it does not seem like the axons start to regenerate. That being said, we do think a cool, eventual path for some of this work is we're able to stop the retinal ganglion cells from dying, but they can't regenerate on their own. There's other work, such as from the lab of Jorgang He, studying these P10-mTOR pathways in order to induce axon regeneration, but they can only regenerate the axons in the retinal ganglion cells that live. And so maybe by combining these forces, we'd be able to both preserve the retinal ganglion cell survival, but then also use these other pathways and methods to induce the regeneration.
Another question is the triple knockout effects, are they specific to astrocytes in the ALS model?
Yeah, so the biggest limitation of the triple knockout model, and the reason we're trying to generate new models as we speak, is that it is a global knockout for these three factors. There are reasons that are somewhat complicated as to why I think there's a good argument to suggest that they are specific to astrocytes in this particular disease model. For instance, in the central nervous system, so we are knocking out the factors microglia normally secrete, and within the central nervous system, certainly microglia are the only cells that express those three genes, but it is true that those genes and proteins have other effects. Often in models such as ALS, the individual knockouts don't have an effect, but the combination does. And the only real pathway we know that requires all three knockouts, but not any individually, is astrocyte reactivity, which I think is an interesting argument for it being astrocyte specific. But that is the number one limitation of the model, and the reason that we're trying to come up with other ways to specifically modulate astrocyte reactivity in a more cell type specific fashion.
So maybe we can take one more question. So what specifically are the toxins from A1 astrocytes?
So that has been the primary push of my entire PhD. It has been a great lesson in how to fail repeatedly in science. I don't have the definitive answer on that. I have some thoughts, but that's the whole other half of my PhD. So probably too long to get into now.
Okay, thank you so much, Kevin.
Thank you.
We're going to move on to our next speaker.